Log on, tune in, find out. Another good idea from Cambridge.
Thank you. So thanks for the invitation to talk to you today about some of the work we've been doing at the Sanger. Genome-wide association studies in complex traits have been very successful in recent years in identifying common variants and underlying these conditions. But there's a substantial component of heritability that is still missing. And several reasons have been hypothesized to underlie that missing heritability, including interaction, structural variation, epigenetics, and the environment. We could be looking at thousands of very small effects at common variants, large phenotype-genotype heterogeneity, or locus heterogeneity, and the contribution of multiple rare variants to these common complex phenotypes. So, low frequency and rare variants can contribute to common complex phenotypes, and there is a growing body of literature to support this. Rare variants can have higher penetrance, contribute to more extreme phenotypes, and may be more useful as predictive markers of disease. And currently, we access low frequency and rare variants either through existing genome-wide association scans, imputation based on reference panels such as the 1000 Genomes resource, or direct resequencing of the study samples of interest. But we know that rare variant analysis can be problematic in that single point analysis, which is what people have traditionally been using for common variants, can be very underpowered. So at an 80% power at the genome-wide significance level set for common variants of 5 times 10 to the minus 8, and then allelic odds ratio of 2, the sample size required for a variant with frequency of 5% would be 2,500 cases and controls, raising to 12,000 for a frequency of 1%, and then to over 100,000 for a frequency of 0.1%. An alternative to these single-point analysis methods is to use multivariate methods to combine information across multiple variant sites across a particular functional region of interest, and that could, for example, be a gene or a regulatory region, or it could be even sliding windows across the genome. Several, in over 20, locus-specific approaches have currently been proposed, including collapsing methods and allele matching methods. But many additional challenges exist with respect to rare variant analysis rather than just the question of single point analysis versus multivariate analysis. And here is a by no means exhaustive list. And the field, I should say, is very active in trying to address these and resolve these. Collapsing methods have been proposed as a way to summarize information across multiple rare variants within, as I mentioned, a locus of interest, here a gene represented by the different red boxes. Every line here represents an individual and every blob a rare variant. And shaded circles represent rare variant alleles. So the first individual carries 2 rare variant alleles out of 10, and so the proportion of rare variants that carries 0.2, and so on and so forth. So one can look at the accumulation of rare variants within an individual and analyze that within a regression framework, and several methods that incorporate this approach have been proposed. We recently extended those methods in Ariel to account for uncertainty arising from resequencing experiments at the variant and at the genotype level. Collapsing methods do really well in the presence of multiple causal variants within a particular locus, but their power tends to drop when many non-causal variants are included in the analysis. An alternative is to use allele matching method, so to to create an allele matching score at every variant across a gene between pairs of cases and pairs of controls, and to compare those similarity scores between cases and controls at each variant and then sum over variants in a kernel-based association test. And again, this is something that we've extended to account for uncertainty in our Amelia software. Importantly, this method is robust to residual correlation between variants within the gene, and also is powerful in the presence of different directions of effect within the same locus. here is a power comparison between the original collapsing and extended to account for quality control quality measures version, and the kernel-based association test and the Amelia. The left-hand panel shows situations where in the simulated data all effects are in the same direction, and the right-hand panel when effects are allowed to go in different directions, so where the gene contains both risk increasing and protective variants. And so in the presence of different directions of effect, the allele matching methods are much more powerful than the collapsing methods. And we also find that accounting for uncertainty increases power, albeit slightly so. We then looked at the effect of sample size on the power of these methods. So the top panel shows the situation of 500 cases, 500 controls in the same, with directions of effect in the same direction and a different direction. And the bottom hand panels for 1,000 cases and 1,000 controls in the same scenarios. So we find that the power of the allele matching methods further increase over the collapsing methods with increasing sample size. And again, that accounting for uncertainty increases power. But taking statistical approaches to improving the power of analysis to detect rare variants isn't the only way to achieve this. One can also, as Richard mentioned, do this by careful study design. And so population isolates have been proposed as a good way to empower the study of rare variants because rare variants may have increased in frequency in population isolates, and a linkage disequilibrium tends to be extended. So one would need deeply phenotyped isolated population samples, and one could carry out whole genome sequencing in a subset of samples and then use that as a reference panel to impute out into the full set of GWAS individuals, and then perform association analysis with the traits of interest. And here's a schematic of that study design, where a population isolate with extended phenotypes is subjected to genome-wide association scan genotyping, and then a subset of samples is whole genome sequenced. And of course, the proportion of samples to sequence is dependent on cost, and hopefully that fraction here will increase to possibly 100% in years to come as sequencing costs decrease. For the time being, detected variants in this internal reference panel can be imputed out back into the full data set, which would allow testing for association with traits of interest, validating robust signals by genotyping back in the original cohort, and of course, upholding the gold standard of replication. I'm involved in setting up one of these kind of studies in a couple of Greek population isolates, and we'll be looking for talented postdocs to join me in analyzing this data over the next few months. So to change tack, I'd like to give you an example of an applied approach to analyzing rare variants using 1,000 genomes imputed data. And this comes from the field of osteoarthritis, a complex disease that's particularly common in the elderly. There are no current therapies for osteoarthritis, and to date, the disease has been relatively refractive to genome-wide association scans, with only 2 loci robustly associated, both common frequency and with small allelic effect sizes. So we carried out a genome-wide scan in the UK across 3,000 cases and 500 controls on the 610K Lumina array, and this is a QQ plot of the results genome-wide with no deviation from the null expectation of no association. When we used HapMap to impute, the picture didn't change substantially, but when we used 1,000 genomes and went down to lower frequency variants, we saw some deviation from the null and prioritized signals for follow-up and replication. And we were able to replicate a particular signal on chromosome 13 across several data sets, all of European origin, for a total meta-analysis of 19,000 cases and 25,000 controls. And the gene within which this variant resides is MCF2L, which is expressed in tissues of relevance in rats. And in humans, the gene regulates neurotrophin 3-induced cell migration. Neurotrophin 3 is a member of the nerve growth factor family, and we know that inhibition of NGF has an effect on the pain experienced by OA patients, particularly in patients with knee-OA, where actually association of the signal was stronger in our data. Finally, I'd like to leave you with a little snippet of analysis of rare variants directly applied to sequence data. And these are preliminary data from the 500 exomes project, which is a collaborative resequencing experiment between the Sanger Institute, GSK, and Lausanne. The study design includes 500 individuals from the Colaus cohort, who all have a cohort of 6,000 individuals, who all have been GWASed on the AFI 500K platform. These 500 individuals all have BMI greater than 25, and half of them have type 2 diabetes and half are normoglycemic. Here are some preliminary data showing different approaches. Single point and collapsing methods tend to be more underpowered, and Amelia shows greater power. And I'd like to leave you, as I'm being asked to stop, with the UK 10K project, which is a large-scale collaboration across the UK to find rare genetic variants in health and disease. And these are the aims of the project, and this is the website. Thank you. One quick question while I go through my acknowledgement slides.